So my name is Alexander Tan, and this year my project is validating Hawaiian stream survey syntheses using environmental DNA and comparing surface depth and side sampling locations for species detections. So, um, wait, okay, so um, my inspiration for my research comes from a few factors. The first being a summer internship I did 2 years ago at the DeWan lab located at UCSF's Proctor Foundation in San Francisco. There I learned how to extract DNA from patient samples, and this kind of inspired me to do more work in DNA involving DNA extraction. And so last year, I did another eDNA stream census project, applying these skills that I learned from my lab. And this year's project is a continuation of that. And the goal of this project overall is to help overall make it easier to help preserve the biodiversity of the Hawaiian watershed and save the unique uh, freshwater goby and also other native species to Hawaii's watershed, including the Oopu pictured here, who are able to scale vertical waterfalls hundreds of feet tall. So yeah, going into this, Hawaii is home to 5 native amphibious fish. And amphibious fish are fish that have a marine larval phase but migrate up through freshwater streams as a part of their life cycle. And this is a pretty unique trait. And all 5 of these gobies pictured here are endemic to Hawaii, meaning they exist nowhere else in the world. So they're very precious in that sense. Unfortunately, due to human activities, the native fish population has rapidly declined in recent years. Um, some of the primary factors include urbanization, which has caused reduction in stream depth and velocity, as well as the introduction of invasive species from the aquarium trade, like guppies, catfish, bass, etc. So this has led to some community organizations making efforts to directly remove these invasive species. And so their work has, since 2015, has amounted to over roughly 70,000 fish, invasive fish from the streams, and has led to a direct contribution of, or direct increase in the um, natives that they've observed in the stream, as well as the decrease in the invasive fish that they've encountered overall. So, um, however, in their works, there are some problems with the way, or there are some difficulties they might encounter when doing these stream cleanups, including the difficulties required to access difficult higher up locations, uh, because they need an entire team of entire manpower team to go and block up the stream and remove each of the fish, or remove the fish from the stream with nets. But that's kind of difficult to transfer multiple or several people, or several groups of people up to higher altitudes. So there may be an easier way I- to monitor the streams and also to identify certain or detect certain other fish that are difficult to find using environmental DNA. So eDNA, or environmental DNA, is an alternative stream survey method that involves and sampling the environment to collect all the DNA that has been left off by organisms in the environment and isolating that DNA in order to detect what species are where in a stream location. So in this case, eDNA is being used for to supplement bio as a supplementary biodiversity monitoring tool. So the research questions for this project were, one, can eDNA be used to validate traditional capture survey data of native and invasive species in the Alawai watershed, which is the Makiki, Manoa, and Palolo streams? And two, which section of the stream bed, when sampled for eDNA, results in the highest species detected? So this means sampling from the same site and rather on the side, the top, or the very bottom depth of the stream and seeing what gives the most species detected. And so for this project, I specifically sampled from Palolo stream at several different sites that these community organizations have also sampled from. And furthermore, to answer the second question, I sampled one of the sites at different locations in order to see which location of sampling produced the best results. So for this project, I after sampling the water, I brought it back to the lab and used a vacuum filtering DNA consolidation protocol in order to isolate the biomass of the stream sample. And then I then preserved the sample using a preservation buffer known as Longmire's buffer. Uh, Following this, I used a DNA extraction kit to isolate the DNA from the biomass and to get just the DNA from the sample. And since the eDNA that we collect is generally in dilute quantities, since it's been in a large volume of stream water, we use a polymerase chain reaction or PCR to create more copies of that DNA so we have more DNA to work with. And once we've conducted the PCR process, during the PCR process, we use MyFish primers, which are general fish primers that enable us to create amplicons or copies of primarily fish DNA so that we can analyze what kinds of fish DNA there are and not other species of um yeah other aquatic species in the streams. So following that, we after checking that the samples were okay to proceed with through quantification and quantification processes, we then conducted library prep and sequencing of the samples in order to prepare them to be sequenced, which is reading the DNA base pairs of the fragments of DNA in the samples in order to help get us one step closer to identifying what fish or what species of fish were in the streams. Finally, once we have the raw reads from the sequencing, we then used a bioinformatics pipeline known as the Dakona pipeline, which involved taking the raw reads, conducting quality control and filtering of those reads, and then clustering similar DNA fragments into an average or consensus. And then finally, running those averages against a database to match the species to the fragments of the DNA fragments in order to identify what fish species were in a particular location. So before I show the results, I got to note that eDNA may be subject to some biases. For instance, due to the general fish primer that we used to create more copies of the DNA, some species of fish are better amplified by the primers than others. So this leads to misrepresentation in the final product. For instance, here the Oopunakea and also the bushy bristlenose plecostomus make up a large portion of the relative abundance from a Nui Nui school sample, but their actual species count was not as high as this might show. So it's not really an accurate way. You can't really quantify the amount of fish of one type of fish there is just from the relative abundance in the reeds. And also, or also some other fish might not produce as much or release as much DNA into the environment. For instance, these smaller fish like rainbow guppies or rainbow guppies might not release as much DNA as like a slimy like Plecostomus. So that might also reduce the amount of eDNA that we pick up and ultimately amplify. So this is a picture of a graph that I made of the species detected by eDNA as well as detected by manual surveys within a time frame of 2 months, 1 to 2 months for each site. And it's a little confusing, so I'll just break it down by results. I found that eDNA detected certain species, including cryptic species, better than manual surveys. So eDNA was able to detect white-spotted Clarius catfish in all locations all 4 sampling locations that I tested. Well, the manual surveys didn't detect it within a similar time frame. And it also detected smallmouth bass, even though smallmouth bass is not usually seen in Palolo stream, but this could be due to contamination. So I'll have to come back to that. And I should also note EDNA did detect some species worse than manual surveys. Like I mentioned before, some fish species that don't release as much EDNA are harder to detect using EDNA surveys. So rainbow guppies and green pterocichlid were not or wait, this is wrong. The rainbow guppies were not detected as well by the EDNA surveys compared to the manual surveys, even though there are plenty of them in the streams. So um, it's it's important to know that EDNA might not be accurate representation of all the fish that are in one location. And as for my second research question as to what is the best relative sampling location, I sampled Chaminade University from 3 different locations from the side, the surface of the stream, and the very depth of the stream. And um, there was very little difference except for one of the native fish, the Opunakea, um, which is Opunakea is a bottom dweller, meaning it likes to burrow towards the bottom of the stream. But the surface sensor sample did not detect Opunakea, meaning that relative sampling location might depend on where your target species are located. So if you're trying to find a specific species, then you should probably look into its behavior before you're deciding where to sample for it. So in conclusion, environmental DNA can be used to verify traditional surveys in some cases, and it can also help try detect certain cryptic species that are hard to capture by manual surveys. However, eDNA probably can't be used completely alone due to its inconsistency, especially to detect low count environmental DNA species, as well as the fact that it doesn't actually remove any invasive fish from the streams. And I should note that neither method for monitoring biodiversity is necessarily without like some sort of error, and they both have different kinds of bias, but together they kind of supplement each other. So that would help. Um, this can help overall the community organization's efforts in trying to restore the biodiversity of the watershed. And yeah, yeah. So some future research could involve creating a complete species map of the other streams in the LA watershed. So um, last year I tried to sample Minoa stream and I had some technical difficulties, so we didn't really um, uh, completely accomplish that goal. So maybe some future students could try to sample the Minoa stream and maybe also Makiki stream and combine that with other streams to get like a complete survey census of, as well as like experimenting with how eDNA applies in stream environments. And also future research could also involve conducting eDNA surveys in collaboration with the community removal surveys in order to increase efficiency of like gathering samples, since I had to gather all these samples myself, like, and it was pretty difficult. So it'd be easier to get them all together. I think that would help any future students interested in researching environmental DNA. Okay, so some advice for future honors independent research students is to not procrastinate. I procrastinated on this presentation, and I think that shows, but yeah, it's important to not procrastinate because the work that you do in independent research will eventually pile up. So you gotta stay on track, and if you do a little work every day, it will get so much easier. I also advise future students to pick a research topic they would enjoy and that others would find interesting too, so that you can share what you're interested in, and other people will also enjoy what you share with them. And lastly, uh, my advice is to be willing to ask for help, because there are plenty of times where I was like confused or unsure of what to do. But Ms. Kobayashi, Mr. Hill, and Dr. Chan, and Mr. Yap, and a bunch of other people I'm about to mention, we're always there to help. So, and it makes things a lot easier. So yeah, don't be afraid to ask for help. Okay. I'd like to give some acknowledgements for people I would like to thank. Uh, First of all, I'd like to thank Dr. Chan, even though she's not here, but she'll probably watch this recording. So yeah. Um, and also Mr. Hill, for your help with my entire project and being my mentors and also guiding me along the whole process. Yeah. And also Ms. Kobayashi for also doing basically the same thing. So yeah. I'd like to thank Mr. Yap and Ms. Shiruk for their insights on the stream, like stream sampling and also their help in getting access to certain locations during our sampling times, and also providing advice overall as to how I could improve my presentation and um, yeah, overall pitching of my research. I'd like to thank my family for always supporting me, and I also like to thank my honors independent research classmates and friends for always also supporting me and being there. And yes, I think that's it. Thank you so much.